Hello everyone, my name is Dr. T.S. Kurne in Department of Statistics, Rajarshi Chhatrapati Shahu College, Kolhapur. In last lectures, we have discussed the what is meant by designs of experiments, their basic terminologies, types of experiments, and steps involving in the experimentation. Now we discuss which principles we have been used in the designs of experiments. So, while designing of the experiment, the following three principles are used. First one is the replication, second one randomization, and third one is the local control. By using the Fisher diagram, the three principles have been designed in this way, and replication is used in all. But if we have used the replication principles with the randomization principles, we validate the estimate of experimental error. If you have used the principles replications and local control together, then we minimize the experimental error. And in any designs we have used three of these principles, then we have valid estimate as well as minimization of experimental error. And in any experimental term called as experimental error cannot be avoided total, because if error is removed in an experiment which is controllable, but which the error beyond the human control, it is chance cause, which cannot be avoided totally. In an experimental error is expected to be minimum. So this chance error which is minimizes but cannot be removed totally. Further, the estimate of experimental error helps in analyzing the results. So we discuss the principles of designs one by one. First principle is replication. In generally, replications means repetitions. So replications mean repetitions of treatments under investigations. And replications in the designs of experiments serves the following purposes. While repeating the treatment of the effect of experimental error is averaged out, hence the validity of estimate of experimental error is considerable. It provides the valid estimate of experimental error. Second, now the replication of the treatment results in more reliable estimates than the single observations, because if we take an experiment and we have take one trial and give the results, this is the not correct results. If we have an experiment, we have take the two trials or replication treatments has repeatedly two times and average out the results, then is the more reliable than the one single observations. Replications also helps to reduce experimental error, and thus it is enable us to obtain more precision estimate of the treatment effects. From statistical theory, the standard error of estimate of mean of the treatment with variance sigma square and the sample of sizes n, it will be sigma by square root of n. This is the formula of standard error. So thus if the r increases, standard error decreases. Means replication increases, your standard error automatically decreases. Results in increase in precision of designs. Here the precision of design is inversely proportional to the square root of the replications. In the designs of experiment, for analysis of variance technique is used. It is mainly used the theory of T or F test. For the validity of this test, certain minimum degrees of freedoms are required, and these degrees of freedoms will be provided by the replications. So next types of principle is randomization. Randomization means randomly use the experiment. So in randomization, allocation of treatment in experimental units such that each treatment gets an equal chance of showing its work. That means randomization is a process of assigning the treatment of various experimental units in a purely chance manner. The following are the main object. The following are the main object of randomization. If we have used randomization, we have removed the human biases or personal biases. So what is the objective? According to the Fisher diagram, randomization along with the replications means this randomization along with replications, we have valid estimate of experimental error. Valid estimate of experimental error. And advantages of randomization in the designs of experiments are, it is logically to allocate the treatment randomly, which helps to avoid a personal biases. And next, validity of the statistical test of significance. For example, T test for testing the significance of difference of two means, F test for testing the homogeneity of several means, depends on the facts that the statistical under consideration obey some statistical distribution. Randomization provides a logical basis for the above test, and it helps to draw a rigorous inferences. Randomization equalizes even a factor of variations over which we have no control. Randomization without replication is not sufficient. It is possible to apply test of significance or only when the randomization is occupied by the adequate number of replications. So next principle is local control. If the experimental materials, say the field for agriculture experimentation, is heterogeneous and different treatments are allocated to various plots at random over the entire field, then the soil of heterogeneity will be also enter in uncontrollable factor and thus increases the experimental error. For the valid interpretation of this analysis of this experiment, the experimental error is expected to be minimum or maximizes. And the use of this local control with replication by using this Fisher diagram, which minimizes the experimental error. In order to minimize the experimental error, the wall of experimental area is divided into homogeneous group. This is also called as a block, row wise or column wise, or both according to the direction of heterogeneity. And the variations within the block is maximizes and between the block is minimizes, because the material in the block is homogeneous. It is a homogeneous group is called as a block. So the variation between the block is minimum, and the treatments are then located at random within each block are perpendicular to the direction of heterogeneity. So the what is the definitions of experimental uniform local control? The process of reducing the experimental error by dividing the relative heterogeneous experimental area into homogeneous block is known as a local control. Thank you.